Hello guys, I am Sachin. Now we are going to see about case study on Azotobacter species. Azotobacter is a nitrogen fixing bacteria isolated from rhizosphere plants. It was discovered by Dutch microbiologist and botanist Martinus Beijerinck in 1901, who was one of the founder of environmental microbiology. We are going to see about general characteristics of Azotobacter species. It is a gram-negative bacteria, oval or spherical in shape, size 2 to 10 ml long and 1 to 2 ml wide. Respiration is aerobic. Optimum temperature 20 to 30 degrees Celsius. Optimum pH about 7.0 to 7.5. Endospore is absent. Capsule is present. Commonly found in soil, water, sediment and plant roots. It is motile. Common Azotobacter species are Azotobacter beijerinck, Azotobacter corcocum, Azotobacter paspali, Azotobacter vinelandi, Azotobacter agilis, Azotobacter insignis, Azotobacter microcytogenes, Azotobacter nigricans. Azotobacter recommended crops like rice, wheat, millet, cotton, vegetables like potato, onion, tomato, brinjal and other vegetables, sunflower, mustard and flower crops. Azotobacter applied field shows 10 to 15% increased yield. Nitrogen fixation. Converting atmospheric nitrogen into ammonia, which can be readily taken by plants. This reduces the need for synthetic nitrogen fertilizer. Biofertilizer. Azotobacter are also used to biofertilizer to improve soil fertility and enhance plant growth and provide a balanced and organic source of nutrient to plants. Soil improvement. Azotobacter improves soil structure, aeration and water holding capacity. They also enhance nutrient availability in the root zones, promoting better plant growth. Crop inoculation. It also applied in the seeds or soil inoculation to specific crops, especially legum, to boost nitrogen fixation and overall crop yields. Environmental cleanup. Azotobacter, Azotobacter detoxifying the soil from the heavy metal and other pollutant. Biological control. Azotobacter produce antimicrobial substance that can inhibit the growth of certain plant pathogens. It is used in integrated pest management. Organic farming. Azotobacter is particularly interest to organic farmer because it aligns with the principle of sustainable and chemical-free farming. Research and biotechnology. Azotobacter serves as a model organism in research related to nitrogen fixation, which has broad implications for understanding the crucial microbial process and engineering nitrogen fixing capability in other plants. Greenhouse and nursery agriculture. Control environments such as greenhouses and nurseries improves soil health and nutrient management are essential for plant growth. Reforestation and land reclamation. Azotobacter plays a major role in reforestation project and land reclamation effort by aiding in the establishment of vegetation in nutrient poor soil. Now we are going to see about nitrogen cycle. First, nitrogen cycle, it present in the atmosphere. It break down by the lightning. It has three bonds. Every time lightning will not help to break down the bonds. So the alternative method is nitrogen fixation. Fixation means converting an element into its usable form. Microbes used to fix nitrogen. Microbes are present in the soil, helps to fixing nitrogen. In soil, there are three compounds: nitrate, nitrates, and ammonia, which helps to synthesize the protein for the plants to grow. The cycle is intake of intake of nitrogen and release of nitrogen. The cycle flows of atmospheric nitrogen to the living world and back into the atmospheric nitrogen cycle. Isolation of Azotobacter species. Martinus Beijerinck was the first to isolate and describe Azotobacter. Azotobacter cells are not present on the rhizoplant but are abundant in the rhizosphere region. Lack of organic matter in the soil is a limiting factor for proliferation of Azotobacter. They depend on the energy derived from the degradation of plants residues. Azotobacter are isolated from the rhizosphere soil by serial dilution method, spread plate method, or pour plate method. The culture medium used for isolation of Azotobacter species is ash base mannitol agar. Microscopic characteristics of Azotobacter. Gram staining. It observed as pink color, oval or rod to spherical shaped gram-negative bacteria arranged in a single chain or clump. Motility. Hanging drop method. Actively motile rods due to flagella. Cultural characteristics of Azotobacter species. In Aspice mannitol agar, the growth of Azotobacter species is large, good and luxuriant. Azotobacter crococum shows brown to black colonies and Azotobacter beijerinck showed yellowish colonies. Now we are going to see about biochemical characteristics of Azotobacter crococum. Catalase test positive, oxidase test positive, urease test positive, indole test positive, methyl red test negative, bogus phosphorus test variable, citrate utilization test positive, gelatin hydrolysis negative, starch hydrolysis positive, casein hydrolysis positive. Now we are going to see Azotobacter species biochemical test. Gram staining positive, motility test motile, catalase test positive, nitrate reduction positive, pigment production positive, H2S production positive, urease test positive, citrate test positive, utilization of carbon source positive, glucose positive, fructose positive, maltose negative, indole test positive. Thank you.